Hello, my name is Carl, and in this video I'm going to briefly discuss the Hardy-Weinberg equilibrium and how selection can affect it. To discuss the Hardy-Weinberg equilibrium, let's first establish what allele frequency is. Allele frequency is going to be the proportion of alleles in a population, so basically the ratio of dominant to recessive alleles. So let's look at the Hardy-Weinberg formula. So we see here p squared plus 2p plus q squared equals 1. Well, so the p's and the q's are to represent the frequencies of an allele in a population. So we see up here a big B and a little b to represent a dominant and recessive allele for the B gene. And we can assign, it doesn't matter which one goes to which, but generally you'll see P assigned to the dominant allele and Q assigned to the recessive allele. And then when you calculate this formula, you will see that the values should equal 1. It's important to note that this Hardy-Weinberg equilibrium equation, it applies to populations in which it is large, the mating is random, and it's not being affected by immigration or natural selection or any other sort of genetic drift. Let's do a fictional example with canine coat color to demonstrate this point of the Hardy-Weinberg equilibrium and how we can test for it. In this example, our homozygous dominant dog has a blue coat, our heterozygous dog has a green coat, and our homozygous recessive dog has a yellow coat. I'll quickly assign our P's and Q's to our alleles so that we don't lose track. Now let's look at the population numbers with 213 total dogs, 8 of them being homozygous dominant, 84 being heterozygous, and 121 being homozygous recessive. Now let's add our total alleles to get our allele frequency, where we have 100 big B's and 326 little b's. Divide that by 426 and we get our respective allele frequencies. Now let's use our Hardy-Weinberg equilibrium values to estimate what we would see as far as genotype frequencies when looking at our allele frequencies. These calculations are showing us our expected number of each genotype in the population according to our Hardy-Weinberg equilibrium. Now we're going to do our chi-square test, where our degrees of freedom are our number of genotypes minus our number of alleles, which is 1. We're going to take our genotype, our observed number in the population, our expected number, subtract the 2, then square it and divide it by our expected number. I'll fill in the rest of this chart, and then we can add all the values in the rightmost column and compare it to our critical value for a 0.05 p-value. We see that it is less than our critical value and fail to reject the null hypothesis and conclude that this is in line with a Hardy-Weinberg equilibrium population. Now let's see what happens when we throw selection into the mix. First, we calculate our genotype frequencies, which I've put here, assign a fitness value to our different phenotypes. For this example, we're going to assume an arid environment where lighter coats will be preferred. We will multiply our genotype frequencies by our fitness value, add all of those together to get an unweighted total fitness, and then divide our values by that number. This gives us our new genotype frequencies after selection. If this were to continue, we would see additional directional selection towards the recessive genotype. Thank you for watching.